Hello dear students. Let's take up a new topic of the chapter Molecular Basis of Inheritance. Till now in this chapter we have already studied about various aspects of the DNA. We studied the molecular structure of DNA, that is the polynucleotide structure. Then we also studied the model which was proposed by Watson and Crick, that is double helical model of DNA. We studied various experiments to prove that DNA is the genetic material. We also studied how this DNA is packaged in a nucleosome. And today we'll be studying about the semi-conservative DNA replication. The method, that is semi-conservative method of DNA replication, was proposed by Watson and Crick only, but they could not give any experimental proof for it. So the experimental proof was later on given by Messelson and Stahl. They performed an experiment which we will study today. And after that, in the next class, we will be studying about the details of the machinery, mechanism, and the various enzymes involved in the process of DNA replication. So let's begin with the experimental proof given by Messelson and Stahl. Let us now study the experiment performed by Messelson and Stahl to prove experimentally that the DNA replication is semi-conservative type. They performed their experiment in 1958 and they used a bacteria, Escherichia coli, E. coli, and they grew this E. coli in heavy nitrogen, that is N15 ammonium chloride. That's an isotope of nitrogen, heavy isotope of nitrogen. So they grew the E. coli in this medium and they found that the DNA is now totally labeled, that is, it is heavy. It's not radioactive, but it is heavy. Now, after that, they centrifuged the E. coli to get the DNA. And they did the centrifugation in cesium chloride. And after that centrifugation, which was subjected to gravitational force, they could get the DNA in the this tube, that is the centrifugation tube, and they called it as heavy because it settled towards the base of that tube. Okay, so this was the first thing. Then after that, they used a normal nitrogen medium. They used the ammonium chloride, but here the nitrogen was not heavy. It was N14 type. Earlier they had taken N15 type. After that, for the first generation, they took ammonium chloride, which was a normal ammonium chloride with N14 type. And then they centrifuged again. That was called as first generation. After 20 minutes, the doubling time of E. coli is approximately 20 minutes. What they found? They found that all the DNA which they extracted was not heavy. It was of hybrid type. So it did not settle at the base. It settled somewhat at the center of the tube, and they called it as hybrid. Hybrid means here both the strands were of N15 type. In this case, out of these DNA, one strand was of N15 type and other strand was of N14 type, which was called as the hybrid. After it, again they cultured it next for 20 minutes in normal ammonium chloride, and then again centrifuged it. What they got? They got that 50% of the DNA was of intermediate type and another 50% was of light type now. So, by performing these experiments, they showed that the replication is of semi-conservative type. Semi-conservative means one strand, the newly formed DNA takes up from the parent itself and one strand from the medium. So in this way, it gets the one copy, one strand from the parent, and depending upon that strand, it can prepare its complementary strand from the medium, from the culture material. So in this way, they explained that this process of DNA replication is a semi-conservative mode of replication. Similar experiments were also performed by Taylor, and he used the flat beans for this. Okay, so this experiment is a very important experiment to prove that DNA replication is of semi-conservative type. This experiment is very important from examination point of view. Please prepare this. Please prepare the diagram. Practice the diagram. Okay. Now let's see the same experiment with the help of a small animation. Now let's recapitulate the whole process with the help of animation. Watson and Crick proposed the hypothesis that DNA replication was semi-conservative, but they could not provide any experimental proof. In 1958, Michelson and Stahl designed an experiment to test this hypothesis. The hypothesis was there is semi-conservative mode of DNA replication, which means out of the two parents, one is the parental DNA which is retained, whereas another template is synthesized from the medium. To prove this, they used the bacterium Escherichia coli, E. coli. First, they grew it in ammonium chloride having heavy nitrogen, that is N15. N15 is non-radioactive, but it is heavy isotope. So they grew the E. coli so that both the templates became heavy. Later on, they transferred this E. coli and grew them in normal ammonium chloride having N14. Here N15 is heavy and N14 is light, which means when they are subjected to cesium chloride density gradient, N15 remains at the bottom, whereas N14 is light and remains at top. So after the experiment, what result they found out, let's find it. So E. coli, first it was grown in N15 media and the harvested cells were transferred to N14 media. E. coli has a doubling time of 20 minutes. So after every 20 minutes, the cells were harvested, DNA was isolated, and it was subjected to density gradient in cesium chloride. So to begin with the experiment, all were N14. After every 20 minutes, that is first generation after 20 minutes, all 100% were intermediate. Then in the second generation, that is 40 minutes, 50% were intermediate, whereas 50% were light. In the third generation, that is 60 minutes, 75% became light, whereas only 25% remain intermediate.